Hi everyone, today we're going to talk about bacterial genomes and genes, but first we're going to talk about just DNA and hereditary molecules in general and how scientists knew that DNA is the hereditary molecule. So I'm going to share my screen and you're going to see kind of infinity for a minute and then you'll see our slideshow. There you go. All right. So how did scientists determine that DNA is the heritable molecule or material of the cell? So in other words, how did people know that there was an actual physical thing that was being passed on between generations that was determining the phenotype of organisms? So we've all heard of Mendel and how he grew peas and looked at their color, their height, the shape of the pea pod, and managed to figure out which of these alleles were dominant and which were negative, recessive, sorry. And but before that, people didn't know very much about genetics. And even after that, it wasn't proven definitively that there was a physical molecule that was being passed down through generations that was communicating this information. So even in the 1940s, it was still being debated as to what the heritable molecule was. And there were many scientists who still believed that the heritable molecule was made up of proteins, because there are many, many different types of proteins in the cell, whereas DNA seems to be pretty consistent. There are only 4 nucleotides in DNA for different varieties of nucleotide, whereas in proteins you have 20 different varieties of amino acids, at least, which are used for proteins. And so there was a very strong coalition of scientists who thought that proteins had to be the heritable molecule, passing on genetic information from one generation to the next. How did we come to find out that it's not proteins, but rather DNA, which is actually the hereditary molecule? Well, first we had to actually definitively prove that molecular inheritance was true. In other words, that there is some molecule, whether it be DNA or protein, that is being passed from generation to generation and providing information on how an organism is going to look and act, whether it's going to be pathogenic or not, if it's a bacterium, etc. So this is a series of experiments by the Griffith lab, or the Griffith team, I guess I should say. And this set of experiments was, again, to determine or to lend credibility to the idea that there was some kind of hereditary molecule that's being passed between generations. So this experiment was done with a strain of bacteria, or 2 strains of bacteria, S. pneumoniae. One strain was pathogenic, and when administered to mice, it did make the mice sick and end up killing them. And there was another strain of S. pneumoniae, which is very similar to the other one, but this one lacked the genetic information for pathogenicity. So in other words, it could be administered to a mouse and the mouse would not get sick. And so then scientists knew that they could kill bacterial cells with heat. And so they found that if they took the pathogenic strain and they exposed it to heat, then administered it to mice, then the mice did not get sick and they survived. Okay, so again, we have 3 different treatments here with 2 different strains of S. pneumoniae. The S strain, which is the one which is pathogenic, S for sick, and then the R strain, which is non-pathogenic. and we know that we can heat kill either of the strains, but in particular, we're interested in heat killing the S strain so that when the mice are administered with what's left over, they do not show signs of infection. But scientists also found that if they took the pathogenic, which was, so it should cells, and mixed that with the R strain, which also should not be pathogenic because it doesn't have the genetics to be detrimental to the mouse, then administered to the mouse, this mix ends up being infectious and leads to the death of the mouse. So again, this is the heat-killed S or pathogenic strain, which shouldn't have any living cells, mixed with the living cells of the R strain, which is non-pathogenic. And again, this somehow leads to the same signs that a mouse would have if it was infected with the live S strain. And so from this, we can take away not that there is some kind of zombie bacteria, but rather that the hereditary information for being pathogenic and making those proteins which harm the mouse were, this information was transferred from the pathogenic strain to the non-pathogenic strain. And even though the pathogenic cells were not living, they were heat killed, that information, those molecules, were still able to be passed to the R strain. So we didn't need there to be any kind of active participation on the part of the pathogenic strain because those cells are dead. It's just the molecule that the R strain was able to obtain and use that hereditary information. Okay, so now we've proven that heritable information can be passed between organisms. We know that it's information in the form of some kind of molecule or something physical at least. So what is this information? Again, is this DNA or is this protein or something else entirely? Well, we really didn't have any other important experiments that addressed this question until 15 years later. And we have the same study system with the mice and the pathogenic versus non-pathogenic strains. And here you could see the actual bacterial growth. The R strain, again, is the one that's non-pathogenic, and the S is, or the smooth, is pathogenic, S for sick. And they just look different when you grow them up in the lab on a petri dish. And what you can do is to take these strains separately and extract different types of molecules from them. So these are all types of molecules we've heard of before: RNA, protein, DNA, lipids, and carbohydrates. So you can fraction different parts of the cell or different molecules from the cell according to density and size, and it's relatively easy to do in theory. And we could take these from the two different strains and then administer them to mice. And of course, whenever we took any of these from the non-pathogenic or the rough strain, it did not lead to any effect in the mouse. But when we took these smooth colony or the smooth strain, we start to see an effect in certain cases. And when we combine those molecules from the S strain, then we combine the molecules from the S strain with the living R strain. So we're not even risking the cells not having been heat killed, as in this case. You might say, okay, well, maybe not all the S cells were killed. Maybe we still had a few that survived. Well, in this case, there's no possibility of that because we're not even adding the cells. We're only adding these individual molecules or these specific molecules. And in this case, we treat mice with each one of these separately. So the first mouse will get the RNA from the S strain and mixed with live R strain cells. The second mouse will get protein from the S strain, and that is again mixed with live R strain cells. The third mouse in the middle there is going to get DNA from the S strain mixed with R strain cells, which is most similar, or what we're expecting to be most similar to the experiment I just showed, and transferring that genetic information, and so on with the lipids and carbohydrates. And so the only mouse which ended up showing signs of infectious disease from the S strain was the mouse that was treated with the S DNA, or the DNA from the S strain that was mixed with the R strain cells that were alive. So in other words, only the DNA of the S strain was able to transfer this information for how to be pathogenic to the R strain cells. The RNA didn't do this. The proteins did not carry this information and provide it to the R strain cells. Neither did the lipids nor the carbohydrates. And furthermore, another experiment was done because some scientists said, well, you know what, it's extremely difficult to actually remove all proteins 100% from a mixture, so we think that you still have some proteins in there, which is a reasonable assumption, except for the fact that the treatment with protein alone did not confer that information and end up with a pathogenic R strain. And so they went a step further and did an experiment where they took the DNA from the S strain and they did another set of side-by-side experiments. The DNA from the S strain mixed with the R strain as they had done previously. That ended up being pathogenic, but DNA from the S strain exposed to something called DNase, which is an enzyme that cuts up DNA and destroys it. When that solution was mixed with the R strain, then the mouse survived and there was no sign of pathogenicity. So in other words, now we have pretty good evidence that this DNA, this molecule, is absolutely needed in order to confer the information on how to be pathogenic from one strain of bacteria to another. And see, this little mouse is the winner because he survived.